First of all, thank you for giving me this opportunity to speak here today. I am Debasmita. I am a postdoctoral researcher in Professor Michael Sharon's lab. Let's start. Have you ever wondered how proteins inside cell teams up with each other, or maybe with the small molecules like drug? Today, I will show you how our novel mass spectrometry methods can help us to look at these interactions in close physiological conditions. We know, like inside the cell, protein forms a dense network of interactions. Suppose we are interested in this protein complex. So what we usually do, we isolate and then do the structural studies. However, in this process, we may lose critical information about their assembly states or maybe weak interacting partners like maybe proteins, cofactors, or even the tiny modifications they undergo after they have made off. To understand the function of the protein, it is very crucial that we study them at their natural environment. In our lab, to address these questions, we study mass spectrometry. So, mass spectrometry is an analytical tool. It has revolutionized how we study molecules in diverse fields, from analyzing planetary samples in physics to mapping the entire proteome in biology. In our lab, we study protein complexes by mass spectrometry. So what we do, we first ionize the intact native protein complex, maintaining all its weak interactions, giving them charges. Then we separate them by their mass to charge m/z ratio. And then we detect them and identify the entire protein complex. As a result, immediately we know about their molecular weight, their folding, solubility, assembly, and many more things. Few years ago, from our lab, we have shown that we can identify protein complexes directly from the human cells. So this method is known as direct MS, which offers rapid analysis of overproduced intact proteins directly from human cell. So here we can see, so our target protein is the Bruton's tyrosine kinase, BTK. Please remember all of my slides. I will talk about BTK. And here we can see the signal from our target protein. So this BTK is a very well-known target, and it's usually used to treat hematological cancer. So after identifying BTK, we also shown that our method is very good in studying their drug interactions. We know that candidate drugs must have a very good cellular uptake, target binding, and high affinity. Okay, FDA approved drug, we know, yes, they are very good in cellular penetration or they have a high affinity. But what about the non-optimized lead compounds, which are not approved drugs? So here we have shown that these two lead compounds, we optimize their interactions, we screen them by our direct MS method. So here we can see, while we are screening, in the case of L5, we have seen only 24% of BTK was bound, whereas in the case of L6, we have seen 75% of BTK was bound. Yes, this is the power of the direct MS method, that it is very good at screening the non-optimized lead compounds, which is very vital for drug discovery. However, we can see it has low signal to noise ratio, because we know that in human cells, we have very little protein production in comparison to bacterial systems. As a result, it is difficult to study their interactions or any modifications. We know, yes, there are few emerging techniques like in-cell NMR or in-cell EPR. They are also trying to understand the interactions, but mostly they can see the probe the changes in the protein structure. In this way, the identity of the bound drug is unknown. And we have seen that our method has this power to screen the drug-bound protein. So we thought of improving the signals of our direct MS by mass shift. So this is the spectrum. We have seen the BTK, which is overexpressed in human cells. So we thought of shifting the mass of BTK by interacting it with other protein. Let's see an illustration, like we can treat it with one protein or few proteins or maybe several proteins, and taking it out from the crowded M by Z region. Also, here, this M by Z up to 4,000 is very crowded because most of the signals from other proteins lie in this region. So this masking our target protein signals. This way we compromised our method's advantage. That's why we want to increase their mass and shift it out from the crowded region. Also, we can see the distribution of the signal across wide M by Z. So the question we asked was, can we do mass shift by self-complementation of split GFP? Self-complementation. It's a very well-known term in biochemistry, where the protein is divided into two parts. One is very tiny, and the other part is the longer. So they both are inactive. When they auto-assemble, they form the active intact protein, which is often we see fluorescence. We chose green fluorescent protein, split GFP, to do our self-complementation. So here we can see the longer part of the GFP, which contains the 1 to 10 beta strands. We see the nice arrangement of the beta strands, and the tiny part, GFP 11, which has only 16 amino acids, the single beta strands. So when they auto-assemble, they form the fluorescent full GFP. So what we did, we have seen the tiny part, the 16 amino acid GFP 11. This tag we have fused with our target protein. So BTK, we just fuse the 16 amino acid part because it won't affect the production of our target protein inside the cells, production or folding. So it won't disturb the target protein. And then we interact with the split GFP. So this is the trick that we are doing, split GFP trick. Also, because of the small size, we can increase the repeats. So we have fused here 3 tags and then interacted with the split GFP. Also, we did fusion with the 7 GFP 11 tags and then interacted and finally formed the full GFP target protein complex. Let's see how we did. So today I will show mostly the results with 3 GFP. We produced the target protein, which was fused with 3 GFP 11 repeats, produced in the human cells. We collect the cells. We have did the lysis, like the cell breakdown. We collect the crude lysates, and then we just titrated this with a purified split GFP, which was the other longer GFP 1 to 10 part. And then go for mass spec measurement. What we see, this we already have seen that BTK, these are the signals of BTK in a very crowded region. So we can at least see this BTK, but sometimes because of the low protein production, we cannot even see these are inside the crowd. Let's see our methods trick. We have interacted this with split GFP, and just see our target protein signal came out of the crowding. If we can see it clearly, the zoomed view, we see there are very few interacting signals with it. So there are very few competing signals. We can clearly say that yes, we have isolated our target protein's peak from the crowded region. And also we can see they have mass shifted. So yes, by this way, by the self-complementary tags, we can induce mass shift and take out our protein from the crowded region. As a result, this approach can be used to investigate different protein investigations. Like here, I have just talked about BTK, but it can be used for any low expressed proteins. And then we can do the studies, any interactions with drug. And so this is the mass shifted BTK, which is very good to study any kind of tiny interactions or their modifications. So currently in our lab, we are studying the drug binding with BTK, and then we will be able to answer more complex tasks in human cells. Thank you. So I would like to thank my supervisor, Professor Michael Sharon, for her support, my wonderful lab members, my family and friends, and also women in science. Thank you.